Soft Genetics, dedicated to the forensic community. In this video, I will be giving a general overview of GeneMarker HID STR human identity software and MaSTR Probabilistic Mixture Analysis software. GeneMarker HID is an NDIS-approved expert system for forensic profiling of human STR samples. In addition to automated size and allele calling, GeneMarker HID has built-in relationship testing and kinship analysis applications, database search tools, manual and automated pedigree construction, paternity trio analysis for paternity index and probability of paternity, as well as cell line authentication, mixture analysis, and an integrated user management system with built-in audit trail and customizable access rights. Reports can also be imported into probabilistic mixture analysis software like MaSTR. MaSTR Enlightened Probabilistic Mixture Analysis software features a rapid and transparent approach to probabilistic mixture analysis, which utilizes your forensic acumen in an easy-to-use Windows environment for research, validation, and casework applications. MaSTR software was designed following OSAC and SWGDAM probabilistic guidelines, along with published probabilistic approaches, and was validated per the SWGDAM guidelines. The analysis code is available on a confidential basis with purchase of the software. Soft Genetics has a long-standing commitment to providing support to laboratories during software evaluation, validation, and training to bring our software tools online in the laboratory. To import your raw data files, navigate to File, Open Data. Click Add, and navigate to your sample files. Select your traces, and then click Open and OK. We can now see that the raw data has been successfully uploaded. Sample files are organized in a file tree to the left. You may notice that my ladder sample is displayed in blue font. This is because in the View Preferences window under the Forensics tab, I have made ladder my ladder identifier. Thus, samples with the word ladder in the file name will be labeled as allelic ladder samples. To zoom in on the sample trace, draw a box from left to right. Scroll by holding the right mouse key and dragging the mouse left or right. Finally, zoom out by drawing a box from right to left. To scroll through the dye colors, click on the dye color icon, or view all colors simultaneously by clicking on the Browse by All Colors icon. To process your data, click the green triangle icon in the upper left, or go to Project, Run. This opens the 3-page Run Wizard. In the first page, select your genotyping panel, size standard, and standard color, or simply select a pre-saved template. Your validation team can easily make templates for your laboratory's SOPs or delete any unneeded templates from the list. Click Next. The second page has a variety of parameters that correct CE artifacts and govern how size and allele calls are made. Marker-specific settings can be accessed through the Panel Editor tool. The default settings are compatible with most analyses, so I will simply click Next. In the last page of the Run Wizard, you can manually select your allelic ladder sample or select Auto-Select Best Ladder. With this option selected, the program will automatically identify the best ladder for each sample. This is particularly useful when using multiple ladders. Check the Auto Panel Adjustment box to have the program automatically align the genotyping panel with your ladder sample. From this alignment, the program will create a new shifted project panel. There are also sections where you can designate positive control templates, adjust the settings for how alleles are scored, and enable options for mixture evaluation or the use of quality sensors. When you have made your selections, click OK. We can see that size calling was successful from the green icons to the left of each trace. To visually review size calls, click the Size Calibration icon. This window allows you to verify size calls at a glance. Here, for example, we can confirm that the pattern for each sample is very linear, as expected. We can also see that the ladder sample is now in bold. This indicates that the ladder passed and was used to create a new project panel that was aligned to the ladder. Furthermore, when there are multiple ladders in a project, the software compares each passing ladder with each sample and selects the ladder that best matches the migration pattern. Allele calls are summarized in the report table to the right, and a similar color scheme is used. A green icon means the peak passed all analysis parameters. A yellow icon means the peak is in the check range for at least one parameter, and a red icon means the peak failed at least one analysis parameter. Table navigation is linked to the electropherogram. Simply double-click on an allele call to be taken to the corresponding peak. This aids tremendously in investigating flagged allele calls. You can pull up more information about the peak by clicking the Show Chart Table icon. Here, among other details, I can see that the quality status is undetermined, and that the quality reason is IMB for heterozygous imbalance and PL for ploidy, which are both common flags for mixture samples like this one. You can right-click to delete or confirm allele calls. Now, after all allele calls are addressed, you can move on to post-genotyping applications like paternity testing and pedigree construction, database searching, kinship analysis, cell line authentication, and mixture analysis. Or you can save reports for import into probabilistic genotyping software like MaSTR. MaSTR requires 3 different kinds of files. First is a genotype file for a reference, known sample, or alternate. This file type only consists of the allele calls. Here are the GeneMarker HID settings needed for this file type. The second file type required by MaSTR is the genotype file for the protocol dataset. This file consists of the allele calls and their peak heights. Here are the GeneMarker HID settings needed for this file type. These settings are also used for the third and final file type required by MaSTR. This is the signal file for the protocol dataset and the mixture file. This file type consists of allele calls, their peak heights, as well as stutter peaks and their peak heights. The 2 protocol dataset files give the program context for peak height variation and stutter, and consist of single source samples, including replicates and a dilution series. The number of profiles needed for this protocol set will vary depending on the lab. Now that we have our files ready, we can open MaSTR. MaSTR has 3 main views. We'll start with the System view to configure the necessary parameters to start a new analysis. Panels contain information about which markers and alleles should be analyzed. You can create new panels within MaSTR here, or there is the option to import new panels as text files, as well as delete any panels that aren't needed. The elimination database is a collection of profiles, usually staff members, that are used for contamination testing whenever the panel is used. The Population Frequencies tab contains population frequency tables for alleles and different markers. You can edit the data by right-clicking, or you can import new allele frequency tables as text files and delete any preloaded options you don't need. The last tab in this view is for Protocol Set, or Standard Operating Procedure. This includes a set of standard parameters defined by a lab to specify variables. The protocol set also establishes the expected parameters for stutter and peak height variation in the statistical profile. The variance factor and list of stutter values can be entered manually here, or calculated using the single source data I spoke about earlier. The profile calculations will result in stutter ratios and variance plots. If during an analysis a profile in the panel's elimination database passes these filters, the result will be flagged as contamination. Once again, you can add or delete protocol sets as needed. Now we'll move on to the second view. A model is required to specify some standard MCMC parameters, like the number of iterations, chains, and whether or not you'd like to utilize stutter information. It may also include some advanced properties and variables to account for things like degradation. The program comes preloaded with a few models, but you are able to import and export models as .json files, as well as create new ones per your validation process. We have now performed the necessary preliminary steps to set up MaSTR. Next, we'll look at the analysis view, which lists all the jobs that are queued, currently running on the server, or that have just finished running on the server. Click New to begin inputting options for the new analysis. Choose a protocol set, population frequency data, and a model. You must also specify some information about the job, a co-ancestry adjustment, name, comments, the number of contributors, and whether or not to calculate likelihood ratios for the person of interest versus their relatives. Lastly, you import the necessary files. A signal file, which is the mixture sample file. Once again, this includes all the peaks, including stutter, and their peak heights. A reference file for your person of interest that contains only genotype information. Optionally, you can import genotypes for known contributors or alternate references to test, like other possible suspects. Press View Signal to see a histogram depicting the mixture sample signal file. Press Submit Job to add the job to the queue. MaSTR can analyze 2 jobs at a time, but there is no limit to the number of jobs that can be added to the queue. While this job is running, we'll click on a finished job to view its results. First, you'll see the summary table showing likelihood ratios above 1 in a shade of green and those below 1 in a shade of red. The deeper the color, the farther away from 1. You can download the summary table here or open the full report. By right mouse clicking, you can also open the full report, delete if you have admin privileges, copy to a new analysis, download the report, or cancel a job that is queued or running. We'll click Open to see the job results in more detail. First are the likelihood ratios. A likelihood ratio indicates how likely it is that a person has contributed to the mixture. You can select between different reference or alternate profiles and view their likelihood ratios across different populations. You also have the option of displaying the LR plot versus random. This plot shows the expected range from 100 randomly generated profiles for each population. You'll notice there is a simple likelihood ratio, which is the likelihood ratio for each marker multiplied together. There is the overall likelihood ratio, which takes into consideration the order of the contributors. Finally, you can optionally list the likelihood ratios for the person of interest versus their relatives. These results can be downloaded as text files. Next up is the Genotype Set Results. These plots are heat maps depicting the weighted genotypes resulting from the MCMC process. This process integrates various parameters like degradation, stutter, and dropout to determine the most likely genotypes that would explain the allele calls and peak heights in the evidence mixture sample. You are able to download a text file for a particular marker or all markers. The first ratio plot displays the estimated percentage of total DNA belonging to each contributor at each iteration, while the second displays a histogram of these same values. Next are the degradation plots, which model a reduction in expected peak height at higher molecular weights. These plots show the estimated values for the different degradation variables as trace plots and histograms. If any non-constant variables are included in the model, a tab for Model Variable Plots will be here, but since this project did not include any, this tab is hidden. The Run Overview lists the parameters supplied to the analysis along with the other run metadata. You can click on a parameter to see a more detailed view. At any point while viewing the results, you can copy the parameters to a new analysis, view the signal file, delete the job if you have permission, and download the PDF report. There are numerous options for customizing the report to suit the needs of your lab. This includes adding your organization's name and logo to the report. The resulting analysis report is a PDF that includes the run overview, table of contents, and any of the tables or figures we've just covered. Thank you for watching.